Okay, so we are going to go right into our emerging scientist session, and we do this session every year as a way to try and get newer people and different people into the field. And so we do an abstract competition, and then we pick a handful of people to come and talk. And so we're very excited about all the talks that we have this year, and they're especially really doing some cool stuff with flow technology that we haven't done before. So the first speaker today is Andres Nevarez. He's a postdoctoral fellow at MD Anderson in Nick Navin's lab. And Andres completed his doctoral degree at UC San Diego, where he was an HMI Gilliam and NASEM Ford Fellow. He was bitten by the imaging cytometry bug at the Sanford Burnham Brebis? Yeah. Wow, good for me. Brebis Cytometry Core. And he used various imaging modalities paired with genomic engineering and deep learning to understand how genomic ripples in the system manifest in the morpholome. So currently, Andres is focusing on integrating the morpholome with other omic technologies to understand how the progression of healthy to diseased to advanced disease. So he's going to share some data and some thoughts about the new imaging cytometers and how they allow us to understand the morpholome. So welcome, Andres.
Hello, thank you all for joining me today. So today we're going to be talking about the morpholome and how we can use these imaging cytometers to really delineate the relationship between morphotype and genotype, this age-old question that we've been trying to connect. But at the heart of this, we need to make sure that this is an interpretable morpholome. You'll see that there's some deep learning things that we need to figure out. And so from the get-go, how do you make sure that it is interpretable? Okay, conflict of interest, disclosure. I don't, as a postdoc, make enough money to invest, but just so that way y'all know. So when the advent of microscopy and pathology happened, most of it was qualitative. And often they were artists. They were drawing what they saw. But that was really limiting us to understand what was happening with the cellular morphology and how this connects to the genomic data. So in the 1900s, we had the phase, pun there for any of our imaging people, of quantitative microscopy and analysis. So first, we were able to start getting better sensors, cameras, and everything else to go along with it, as well as the automated image analysis and computer vision. Now in the 2000s and past, let's say like 5 years, we have this era of imaging cytometry assisted with this AI, all these advanced models that will allow us to extract more data from the cell. And that brings us to single-cell morpholomics. Single-cell morpholomics is inherently quantitative, and it's the analysis of cell morphology, but it's not just size and shape. Everybody focuses on size and shape a bit too much. We need to investigate other things that you can get from these algorithms or deep learning, such as structure and organization of the cell. So there's two arms to this. You have morphometric features on the left. These are human constructed algorithms that describe the cell. Things like cell shape and size, texture, light scattering, and these spatial distributions of organelles are really just the milieu of the cytoplasm. We have algorithms to describe this. Now, on the other side, we have deep learning features, but these are very nebulous. It's a black box, and you're trying to interpret these. Often, you're doing it after you have your classification model, but that's limiting us in terms of what changes in the morphology actually mean something. And the deep learning features are often these latent features or these embeddings from the model. So I wanted to put this up here. Not all of these papers are going to be imaging cytometry based, but there are bioimaging papers that connect the image features from the nucleus, the membrane, intracellular organization, and even metabolic alterations just using different imaging techniques. So there is a real connection between morphology and cell physiology, and we can track these changes in disease and gain information about this that we didn't have before, beyond the EMT, the classical thing you see in cancer. So beyond that. Now, we are at a point where we can now sort based off of morphology. And ideally, you have these UMAPs based upon whatever algorithms you're using, combination of morphometric or deep learning. But the idea is you identify clusters based off the morpholome, and you want to sort those to do your fun stuff afterwards. So this is done in suspension. There's two instruments that I was able to use. The Deep Cell REM-I, which is a bright field, completely label-free, and the S8, which is also label-free, but then has some fluorescent channels as well. Okay, so I was the Spring Science Challenge winner for Deep Cell. So we were able to send some samples and get preliminary data. That's what I'll be showing you today. And we have it finally in the lab. It took a year to get it with this AI cybersecurity at MD Anderson. But we finally have it in the lab. We're doing experiments. Things are going well. And we're now at the point where we're going to be doing some really cool things. And I hope to share that with you next time. I'm sure people are familiar with this, but just to get us on the same page, we have a single cell suspension. It's a chip. It's a microscope in the black box, 63x bright field, and you're imaging these cells. Each image is sent up to their cloud, and then based upon that, you can make a decision of who gets sorted into what well. And it is based off of the human foundational model, is based off of both morphometrics and deep learning features. So it's combined to make a profile of the morpholome for each cell. Now we run into the issues though, is now we're incorporating these AI features. Well, when we look at our cells, and these are images taken from our pilot deep cell experiments, this is clustered, it's a UMAP clustered off of not patients or cell line, but off of the morphotype. And so we're using light and clustering to do so. Now you see these montages, you can see differences there. Some of them can be quite subtle, but what would really be great is if you have to be able to account for the heterogeneity between patient samples that we're ultimately going to get, and how do you visualize what the spaces, the variance for these morphotypes. And that's where we're going to come in at the end to talk about generative AI models to help us understand a lot better than just montage pictures. But what are these top features that are defining these different clusters? All of them are deep learning embeddings. So that's going to be a possible problem when it comes in terms of interpretation, but it's more accurate in finding these subtle differences than we can find. So here's some data. We have an MCF10A, which is a normal breast cell line. These are heat map UMAPs. We have MDA MB231 parental, and from that we isolated physical single cell clones and grew them up. They're all at the same passage number, the same densities, and then these were sent out. So we're seeing that these 4 clones can recapitulate its parent space, albeit in different spots. And this was what we were not really expecting. I didn't expect C1 to be highly localized to an area, and then C28 being highly localized. But there's some overlap, but it's not to the same probability space that a cell would occur. What is interesting too is that this 231 parental, it's seen in the other ones, but not at the degree it's seen in a parental. And then you kind of have some shared morphology space between all samples. So it could be this memory from normal breast cells to a triple negative breast cancer cell line. So let's switch to the BDS8 with CellView. All of you are familiar with it. I'm just going to bring it to here for these imaging features so you can sort off of these morphometric features. And so you can identify this. So what I had done is just image. I imaged to see what are the features that are going to then define, allow me to discriminate between different populations of cells. So let's just image. And some of them are the same samples as from the deep cell experiment. but I'm not going to show that direct comparison, but I'm open to talking if we want to talk about that. So the BDSA cell view, what I had done was MCF 10A, same cells, the 231s, and SKBR3 are the HER2 altered cell line. So I had stained for mitochondria, and then we have the bright field forward scatter and side scatter to determine. Now, I didn't just focus on the 15 or so morphometrics that they use to sort. Everything can be calculated after the fact. So you have all these images, and then you can calculate all your morphometrics for each channel. So these are all 4 channels, all morphometrics, and reduced when using fate mapping, similar to t-SNE or UMAP. What we're seeing is an axis here shared between the triple negative breast cancer cells, the dark green, and these SKBR3, which is more HER2 status changes there. Along this axis, though, you're seeing these dark spots, and it's similar, albeit different spatial organizations. And you're not seeing the MCF10A normals in this axis or at very low amounts. They're shifted over. So just based upon these channels, you can get enough information to classify these different cell types. And now we have the features that we can then sort off of. But what we also want to do with these images is run it through a classification model and then take that that's convolutional neural net. You're going to have a piece of that, and you can then use that to project into UMAP space and see if there's classification differences. Unfortunately, the codec is unavailable, but the movie it was really, really cool. It's a 3D animation of a t-SNE plot morphing from iteration 0 to our final iteration. So imagine this moving, coming at your face, and then you're super blown away. Awesome. Okay, but now this is the real important part of it. We have these technologies, but we need to interpret, and we need to understand, because as biologists, we need to generate hypotheses. So this is going to help us using interpretable morpholomics. I'm sure you all have seen this from Deepometry and Carpenter and her group over there, but I show this so that way you have a cell, you're entering the network, you might catch edges in the first few layers, then you're going to catch things such as texture or differences in shape. But as it goes on, it's more and more less interpretable. So let's open that box, but how we need to change the model from the beginning. So this is a paper that came out. It's looking at these wild type cells, and then it's a cancer mutation that's enriched in metastatic melanoma. The top row is light scattering, and the bottom row is the shape. And so as you move into this metastatic space, you're having increased in the epithelial shape and increased light scattering. And this was actually corroborated by another paper that's using a generative adversarial network. So you have the original image, and then you create a morphing of it. So we were actually able to differentiate between stage 3 and stage 4 metastasis. So the information's there, but now that was all done in 2D. How do we apply this to these other imaging modalities that we have? And I don't want to spoil everything, but we do have some really cool, we'll call it explorers, so accompanying software that can go along, published, that can go along with these imaging cytometers, and it'll be agnostic to system. Give us images and we'll help you explore the space and then connect it to what other omics that you have. So I want to thank the NAVEN Lab, the IDSO Fellowship, the Deep Cell Grant, and the patients. And so it's really important to orient ourselves while we're doing it. So thank you for your time, and I'll take any questions. Hello.
Hi. Wonderful. There was someone else? Oh. Wonderful talk. I was wondering if I understood well, if you can use the typical fluorochromes for cell classification in combination of this, or if you have found cases that the morphological markers, if you want, classified differently than the typical flow cytometry markers. If you have found those kind of whether they are complementary first, or if you are you find contradictions.
Right. I am not doing any traditional markers for that. One of the reasons is, I know this was in cell lines, but we're getting patient data. And so the tumor samples, they have to be processed very quickly or they'll start to die. And then you get into these caveats of, well, what does the morphology mean? And so that's why we're focusing on this label-free. And because when we've done traditional studies, we see even the gene expression is completely changed. And so it's like a variability issue and a batch issue. And so with these systems, there's more consistency in terms of the background and leads to less bite from the early stuff. But other contradictions between the morphology markers and yeah. Thank you. Thank you. Oh.
Yeah, hold your applause. Thank you so much for showing that. I work at BD, so it's really exciting to see some of the S8 images being used. It looked like you had a fluorescence channel as well for mitochondrial.
Yeah.
factor. Okay. Did that like clearly or not clearly like add anything to the ability to classify here when you're comparing to the purely label-free?
So I did the holdout. Yeah. So I first started with just the bright field. Bright field was like 23% accuracy. And then incorporating each one, you did get more. The highest increases were the side scatter and the mitochondria. And then it would be certain things such as like image moments or things within there. Shape is going to be a bit homogeneous because you're putting them in suspension. So we need to go beyond that.
Yeah, for sure. And so, in this amazing video that we all got to imagine, was that using the image features that are provided from the S8, or was this using features from your classification?
Using a CNN feature, so
Okay,
the
so CNN,
convolutional
yeah.
neural nets feature.
Yep.
So one of the last layers, you then take that information and then project it on here.
Okay. Do you mind saying what the dimensionality of that final feature vector is?
Oh, that
order of
one
magnitude?
is 50, and
Okay.
so it's a resonant 50 model.
Okay. Okay.
And so so then we can get that information from the penultimate layer.
Awesome. Yeah. Okay. Thank you very much. Now we can.
Thank you very much. That was great. Huh? No, classic us. Are you threatening me? Okay. Our next emerging scientist is Alyssa Fears. Alyssa received her PhD in biomedical science and a master's in public health and tropical medicine from Tulane in New Orleans. And currently, she's a research scientist at the Galveston National Lab at UTMB, focusing on early disease biomarker discovery for high consequence pathogens in non-human primate models. So the non-human primate work is in a high containment setting, and it necessitates multiplexing as much as possible, as samples are precious and storage space is incredibly limited. Driving to get the most out of every sample, Alyssa has found a passion for designing and deciphering large data sets, including bead-based immunoassays and high-parameter spectral flow. She just recently joined ISAC and has since also joined the Biosafety Committee as an observer. So welcome, Alyssa.
Thanks, guys. I'm really excited to be here and able to share this 41 color spectral flow panel that we've developed for non-human primate tissues, and we're going to be focusing on PBMCs today. So why on earth would we want to actually start creating such a huge panel? And I'm actually going to start my timer so I don't go over. So we work with 3 major non-human primate species: the rhesus macaque, the cynomolgus macaque, and the African green monkey. And we use these 3 to compare. We do model comparison between these 3 we will test new and emerging infectious diseases in these models and hopefully be able to look for biomarkers in them for human disease. We also do a lot of vaccine and treatment studies, and we want to look at antigen-specific responses and functionality. So a panel like this gives us the ability to broadly and deeply immunophenotype. And so that's why we kind of decided to go for this very large route. I'm going to talk to you today about some of the different things that we've done to optimize this panel. And first is reactivity verification, because reactivity with non-human primate species is slightly different than human. So we need to make sure that our antibodies are actually binding to what we're expecting. Then we go to fluorophore selection and antibody titration. You guys have seen all of this before. I'm just reiterating things that you already know, but we're going to see some examples. Then I went on to surface-only staining for the panel, then added intracellular staining back in, and kind of flip-flopped until I was done making some revisions. So first, we're going to go through reactivity verification. On the left side, you'll see CD19 and several different clones for CD19. The only one that we found to be reactive for African green monkeys has been the J3119 clone from, I believe, Beckman Coulter. I did an in-house conjugation for Delineate 350 that obviously was not bright enough to be able to get good separation. And then their PE Sci5 is commercially available, and this is what we ended up using for our panel. And the HIB19 and C19 clones are not reactive. On the right side, you'll see TCR gamma delta. I bought them all in PE first to see if the B1.1 that we've used previously or the B1 would be better suited for our purposes. The B1.1 is reactive, and it's lightly more reactive for African greens here. You'll see from the separation, but I can actually increase this separation with titration. So we ended up using the B1 clone, and it's we got it from BD. This is the only custom conjugation that we use in the panel. So next, we'll talk about fluorophorochemistry and stability as it relates to this panel specifically. As I said before, in pink here, you'll see TCR gamma delta. That's on RB613, I believe, and that's a custom conjugation so that we could get our best separation here. And I'll also direct your attention to CD107A down here. It's a degranulation marker, and that needs to be added during stimulation. And so that antibody and fluorophore conjugation needed to be stable at 36 degrees for 4 hours for us to be able to actually use it during stimulation. And so this is on, I believe, and I had it in my notes, but I can't see them. I think it's APC Fire 750 to be able to use that. This was, again, designed for the Cytec Aurora 5 laser, and the complexity index overall is 22.46, which is pretty good. Let's see if that goes away. Perfect. So we did do some antibody titration, obviously, for all 3 species. This is a very common question that I get. Do we need to use different titrations for all of the different species? As you saw for TCR gamma delta, kind of, but you can still get good separation. It's just maybe not the best separation. So you can use the same cocktail for all 3 species if you would like, but you won't be compromising too much if you decide to use a single cocktail. Next, I'm going to go on to stimulation and unmixing, which are both also very important. One thing that most of you will note is that if you use PMA and ionomycin, you'll get internalization of CD3. Not great for immune phenotyping, and so we actually use pokeweed mitogen instead in our lab. I've also found that it's a bit more realistic when you're looking at cytokine responses. So if you're looking to get a good cytokine response and make sure that your the brightness of your fluorophore is correct to be able to actually detect that, you might be blowing it out of the water with PMA and ionomycin. And so pokeweed mitogen is much more realistic, and that's what we use in our lab. And additionally, because we talked about DCs yesterday, I just wanted to point out that we can detect this very, very small population. It's about 0.25% of our cells, and you can still see your cytokine responses with those. And it's also broadly activating. So on here, I have both DCs and T cells. So lots of revision. So I first started with CCR7 on SparkBlue 574. It's not very bright. Yes, it was a bad idea, and I kind of knew it at the time. We had Spark YG 581 in our lab already. So I ran both together. You can see not great separation and not great staining for the SparkBlue 574. It's great for other antibodies, just not for this. But I did get some pretty good separation with Spark YG 581, but I really did not like this negative spread that I was seeing. So in the final panel, we use APC Fire 810. I get really good separation, and I don't also get that negative spread at the bottom. All right, so the overall staining protocol is something that you guys have probably all already seen before, but I do want to reiterate that we're going to be stimulating for 4 hours in the incubator with peptide or pokeweed mitogen as our positive control. And again, peptide for antigen-specific responses. Also with Golgi plug to prevent cytokine secretion, and CD107A for degranulation. Then after 4 hours of stimulation, we do our what I call pre-staining, viability staining, as well as blocking with 2% mouse serum. There isn't really a good FC block for African green monkeys. And so I'll give a plug for Calibri Cytometry, Oliver Burton's blog. He did a really, really great job of doing a blocking series. And so I've incorporated some of that into this panel. So blocking, then rare or serically hindered antigens. TCR gamma delta is very well known for needing to be stained first. CD14 as well is not very reactive. The only clone that is reactive for African greens is M5E2, and it's still not great. So I can boost a little bit of detection by adding that first. And then CD27 and CCR7 are also some problem children that we've added to the beginning. Then after those have been stained for about 30 minutes, we'll do the rest of the antibody cocktail, then permeabilize and fix, and then do intracellular staining. And yes, this is going to be at BSL-4, so the fixation is very, very important. Then this gets run on the Cytec Aurora 5 laser, which is at BSL-2, and we do data analysis. So in the interest of time, I'm going to put this up here. I know that this is recorded, so you can screenshot it later. I just wanted to show you that we can definitely do this. And that CD1C positive CDC population is about 0.25% of the 1.5 million cells that I started with here, and you can still see our TNF cytokine here. Again, this is another screenshot for later, but on the right is the workflow that I use to be able to create this UMAP, and you can see there are differences between cynomolgus macaques and AGMs. Rhesus aren't on here just because it was a lot to put on one slide. And then I have stimulated and unstimulated, and you can see definitely differences between both species and also with stimulated and unstimulated cells. And we've got our major players. I went back through and looked at all of the markers and kind of given you some orientation on where these are on this UMAP. So I did want to take my last minute to talk about the importance of having really good tools. These, if you have an Aurora, this is probably pretty familiar to you. It's a screenshot from SpectraFlow, and this is RB545, which CD16 is on in this panel. And if you look at these normalized spectra, they don't look very different, yet I am seeing a difference in the unmixing if I use, this one is just the beads, and these are E-beads from, E-beads plus from Thermo Fisher, or the SpectraComp beads cell mimics from Slingshot. And if I use cells, I get really good unmixing. And if I don't use cells, if I use E-beads for this particular fluorophore, I end up getting a big long row of unmixing errors. But you can't see in these normalized spectra where that's coming from. But if you throw it into R and you plot all of these normalized spectra individually, you can see that there are some changes within where the Virebright V515, which is on the bottom over here, you can see that there are some changes right here, which would explain this. But I would never see that before if I didn't put this into R. So just your tools are very, very important to help you figure out what's going on. Though also, if you listen to your FAS, they're going to tell you if you have some unmixing errors, why don't you use cells? And that will also fix it. So, but this, you can see where it's coming from here. So that's really cool. I did want to give a shout out to the lab. You guys are awesome. And also to our reagent reps, because panels like this are not possible without them. And with that, I'll take questions.
Success. Okay. Very nice job. I'm sure it's very unsatisfying to condense such long work into like 10 or 15 minutes,
Yeah.
but I'm sure everyone here can appreciate it. One question I had was with non-human primate panels, we can often achieve cross-reactivity with human antibodies if we just kind of bump up the volume. So first question was, did you have to do that in this panel? And if you did you run into any issues with antibody aggregates or the loss of signal or loss of resolution, things like that? And were you able to mitigate that with any kind of commercially available reagents or just trial and error with like sequential staining?
Yeah, so sequential staining is really big here. Like I said, the CD14 clone is pretty good, but for African greens, it's not great. So I did have to bump that up and do sequential staining for that. The CD19 clone is also, again, like it's the only one that's reactive, but it's still not great. And that's another one that I've talked to the reagent reps and said, for non-human primates, we really do need better clones. CD38 is another one that's in this panel. It's probably the weakest link. It's on Alexaflor 488. It's from Santa Cruz, and it's the only one that you can get. I could try to do a custom conjugation for that as well, but it's a little bit more difficult to get that from one vendor to the other, and it's a much more difficult process. And I wanted this to be something, we're hopefully going to submit this as an OMIP so that everyone can use it. And I wanted this to be something that people can just order from, whoever their vendor is and be able to throw it together.
So we've got our final emerging scientist. Okay, so our final speaker is Jamie Tijerina. She's a PhD student in the lab of Alexander Chase in the Earth Sciences Department at SMU in Dallas. Her research explores interactions between the environment and human activity, investigating them in the context of health outcomes and environmental economics. Jamie began her career in flow cytometry in 2010 at the Cytomics and Cell Sorting Shared Resource Lab at UPenn. And most recently, she served as a scientific researcher in the flow cytometry and cell sorting facility at Caltech, where she specialized in non-traditional sorting, including bacterial and submicron sorting. She earned her Bachelor of Science in Biological Science and her MBA from Drexel University in Philadelphia. And in just a second, she'll be coming down to talk. But please join me in welcoming Jamie.
All right, so there's Diva, and then there's the other Diva. So there's no presenter mode on the screen. So give me a minute. All right, so we spent a lot of time on this slide. So, okay. Let's see. Let me get used to this too. Just click it. All right, I have to click the, no, I got it. All right, so this is probably not where you thought I was gonna start. So I figured we were gonna have a lot of technical talks, and then I was gonna say a lot of technical things. So you probably wanted a mental break. The occultist movement in Los Angeles and Pasadena runs hand in hand with the early days of Caltech and JPL. Jack Parsons, one of JPL's founders, which also is appropriate because we just had somebody from NASA, was deeply involved in this world. So I'm leaving you with a quote from one of the producers of the series they filmed on campus a few years ago before the pandemic, and a list of things that you can probably just Google and go down the rabbit hole during the break. But this is not just trivia, right? So it's a reminder that many groundbreaking ideas were once ridiculed or misunderstood. That same skepticism surrounds submicron sorting today. All right, so now that we've covered some locational context for where the research was done, I'll continue with broad examples of a few types of submicron samples that you may encounter, or maybe not, I don't know. Plus a dot plot from one of the actual submicron sorts I did. It was actually the last sort I ever did before I left. So anyway, you can see that things don't look so different from any other sort that you would do on a regular day on any other samples in DIVA. That's because I obviously didn't invent submicron sorting, single cell, single virus, single vesicle sorting. It's all the same technology. It's nothing new. So it's just really a matter of optimizing the protocols and procedures for specific applications and instruments, plus actually be willing to accept and do the experiments in the SRL. All right, so if I'd asked people what they wanted, they would have said faster horses. So the basic technology, again, behind flow cytometry and sorting has not changed very much. People have been sorting viruses and microbes, organelles, tiny things since at least the 70s. What has changed is that in the post-COVID era, which I consider to be sometime around mid-2021 when they started making everyone go back and kind of changing the rules towards the return, which that was a thing. That's why there was 2 pictures of COVID in that other slide. So what has changed essentially is that I've been to a lot of courses, I've been to a lot of conferences in that time, and everyone in SRLs is just anecdotally saying that the experiments are getting weirder. And like, you know, it's good weird, right? Like, no offense. But, you know, this is coming from me, where almost half my sorts are non-traditional sorts, non-traditional species. Weird doesn't just mean new species. It also means more challenging emerging applications outside of strictly immunology, like synthetic biology, microbiome analysis, and more. So new sorters have the same limitations as the old for submicron applications. On a micro level, the instrument companies, they're building faster horses. The shift from PMTs to APDs theoretically offers advantage for submicron detection, but in practice, the results have been inconsistent. Small particle detectors are not mandatory for submicron sorting. Spectral instruments don't inherently improve sensitivity for submicron detection. That's just not the nature of spectral technology. And while software tools can offer insights into the instrument sensitivity, the stochastic nature of submicron particle detection limits their reliability. And on a micro level, SRL researchers, individual labs, and independent scientists are also building faster horses. You have to work with whatever is on the market or whatever is in the lab and work within those constraints to deliver results. So I bring up the fact that even though this quote is famous, especially in the context of business, innovation, and entrepreneurship, it's actually a myth. Whether Henry Ford truly said this can't actually be verified. And given everything I just said, that's the big question that many people have around submicron sorting, which is, are the results real? Are you truly sorting the desired target? And can it be validated? And yes, but I genuinely believe that both the sorting and the validation are much more qualitative in nature. And I think that's okay, right? I think imaging technologies and cultivations are key techniques for the validation. So I'm gonna start out with 3 case studies of microscopy validated examples. And then I'm going to move on to the operational context of the SRL and the considerations and challenges doing these sorts in a general SRL setting alongside more traditional experimentation. All right, so for this sort, we used a newer, really bright DNA stain called Miami Yellow. So Miami Yellow was optimized for use in established cell lines, things you see usually like the hex cells. So none of Tyler and Karina's peers outside of Caltech had used flow cytometry in this system. everything is novel, including the use of the stain. So we tried Herxt and propydium iodide. Those xenon dyes, neither was suitable because they were very dim. And also chlorophyll is autofluorescent in the same channel as the propydium iodide. Although Karina was later able to remove much of that chlorophyll in her preparation. So we optimized the concentration of the Miami yellow on using both the SY3200 sorter that we have, it's by Sony, it's an old Jetner sorter, and the Cytoflex S cytometer. And then we proceeded to the sort. So the limit of detection on the SY is 0.5 micron. And this was determined with extensive testing and optimization with help from our service engineer, Mike Pahalja. So this was a proof of concept pilot sort for a graduate student developing their project. So work with mini cells is often in the context of delivery of therapeutics and synthetic biology. So I knew we'd be able to see the sort and Tamaja's sample on the fusion. So I was able to characterize and test the instrument to sort down to 100 nanometers reliably. And so we sorted about 400,000 mini cells for validation by microscopy. In addition to instrument preparation, we made sure to have the appropriate procedural controls to determine the settings and gating strategy. So of course you need buffer-only controls and then buffer and stain with no sample. And then you can try a negative control, but if you're looking at something that's so small that you need to trigger off the autofluorescence, then you either need to characterize what channel that autofluorescence in is and try and trigger off that channel and then use the gating from that as kind of a negative, especially if you have a strong enough positive that's either in a different channel or in that same channel, but much brighter. Or you're gonna have to rely on the previous procedural controls as negative, so the noise. Then you're gonna have to dilute the sample to achieve the appropriate flow rate for the sort. So this is hard because when you're at the limit of detection, more particles can be going through the instrument than what the instrument can actually detect. So even with the low flow rate and the low event rate, you might still experience clogging of the nozzle. So that's fun. All right. So flow cytometry is a cultivation-independent technique for microbiome analysis to evaluate both human and environmental samples. So existing knowledge around evaluating microbiomes in different systems is limited by the fact that many microbes are not suitable for culture. So the idea for current experimentation, very broadly, is to understand community dynamics, characterize previously unknown species, and examine their relationships to situations like climate change, disease, and more. So instrument sensitivity is important to obtain the most representative sample that the existing technology will allow and ensure that as many viruses as possible are detected since they are found at or near the limit of detection. So I wasn't able to use sorter protocols that had been previously developed to do iterations of this experiment on an influx. So we developed new protocols to prepare the BD-Faxaria fusion and another sorter as well. So Francisco worked on optimizing his sample prep to get the brightest fluorescence possible, and this is key for improved detection as well. So before sorting for genomic sequencing, we sorted for validation by microscopy. So panel design is important even when you only need 2 colors. So the choice to use PA647, which fluoresces red, and Cyber Gold, which appears green, made validation by microscopy much more useful. The downstream sequencing requires whole genome amplification due to the small quantity of DNA that is found in viruses. So the protocols to prepare the instrument to sort for that application also needed to be DNA-free and low endotoxin. So because so many different BD sorters, including the S6 and the S8, share the same fluidic path, these protocols will remain very important and useful into the future. All right, so here's some of the operational context. So many aspects in the facility that I was in, Caltech, are common to other SRLs, but a few things here are different. So Caltech has a BD-Faxaria fusion and a Jet and Air Sony SY3200 plus 2 analyzers, one of them being the Cytoflex S and the other is a Miltenyi Vibe, and they're pictured here. Sheath fluid is entirely made in-house, and so that's no surfactants and no preservatives. The only one that we don't do it in-house for is the Vibe. There are multiple operators in the facility, so just like any other facility. But I've shared just my sorting stats from basically, again, the post-COVID era, just to highlight what sets the facility apart in terms of the experiments that come into us. So I personally handled all of the submicron sorting and microbial sorting. So in total, 45% of my sorting only is non-traditional applications. So typically the recommendation for submicron sorting is that only one operator runs the instrument and that it be dedicated only to those experiments. But in most SRLs, that's just not realistic. So what makes this facility stand out is that we accepted the experiments and found ways to work around those challenges. Caltech offers primarily operator-assisted sorting with a few exceptions for the fusion sorter, where the bulk of the submicron sorting is done. So you have multiple operators in the facility, and then you have trained users from outside also using the facility. So all instruments are under service contract. That's really important. Service engineers are key colleagues in this work. And so long before the pandemic, I'd established my own protocols for cleaning and testing and instrumentation for general sorts, yeast and microbial samples. So having that baseline to work with was already really important. And then of course, beyond time and patience, there is the economic reality that purchasing a new cell sorter is a costly risk that many individual labs and SRLs just simply can't take. We did purchase a new instrument, but it's not pictured here because I know there's a lot of clinical folks out here, and we protect the identity of the patient in any good case study, right? So I know that testing the instrument takes troubleshooting and extra staff time, which is a shared challenge in every SRL, regardless of the sample type. But in submicron sorting, the added risk is that the instrument is not guaranteed to outperform the existing ones, despite theory around newer instruments with APDs being more sensitive. Nothing comes without exchanges and compromises. And then of course, theory versus practice. And then finally, I want to highlight my co-author's contributions. So he provided environmental samples and spent a lot of hours helping test instrumentation and providing feedback on results. So testing with seawater samples specifically provided a safe, ethical, and effective way to evaluate the new and existing equipment using BSL-1 biological samples instead of just beads. So in your testing with environmental samples, the results that will work for both the results in those protocols will work for both mammalian cells and microbial cells. It's not typically done in SRLs, and the companies that produce the instrumentation usually only test with mammalian cells or beads. But you end up developing much more robust maintenance procedures. And then when somebody does come in with microbial samples, you don't have to stop and generate new cleaning protocols. You already have them ready because you did that testing with those kinds of samples initially from the beginning. And then also too, when you have things that go wrong with an actual sample versus just running beads, you'll be able to catch those things because beads are always going to be perfect, right? They're beads, they're manufactured, and you just put some PBS or PBST in them depending, and everything's fine, right? So this was very important. All right, so finally, some considerations and challenges. So mindful scheduling has always been really important to make sure everyone gets their work done and minimize the downtime for cleaning. But scheduling in an SRL isn't just about time management. It's also about balancing, unpredictable or novel experiments, instrumentation doing its thing, as we said, diva, and of course, operator workload. Our only work is not just operating the sorter, right? So this is, why a conscious, mindful scheduling methodology works. Also, the necessary instrument preparation is mentally and physically demanding, and burnout in SRLs in general is another topic. It's beyond the scope of this talk, and I'm gonna run out of time soon. But, I also really quickly want to address, maybe not that quickly, address professional development. So normally I would keep it much shorter, but given the moment I think we find ourselves in with research, I think everybody knows what I'm talking about. I don't have to go into the politics here. But ultimately things are, not just a job, right? It's your career. And there's a difference. So it's the future of the work, if it matters to you, and the impact it can make too. And so engaging in training and continuing education, it's an investment in yourself that helps others, and it improves your career stability in a climate of layoffs in industry, threats to science, and then of course the always and even more precarious nature of research funding. So what we did was we scraped together funding so that I could attend conferences and classes. And so this included applying to scholarships, travel awards, putting in my own money. And I also read the literature strategically and then identified the knowledge gaps that exist with the theory and the practice. So whether your employer can fund it or not, the professional development is key, and whether it generates the billable hours for the SRL. So what's unique and why I made it up here, and thank you again for having me to speak here today, is the combination of all these considerations in the course of the day-to-day work. Everything about SRL research and submicron work is interdisciplinary. So it's how we took the time to do the R&D in spite of the fee-for-service funding situation that all SRLs have to deal with. We researched the landscape of the market for sorters and reagents. We accounted for theory from various fields to produce usable results, and then collaborated meaningfully with investigators to gain the insights into the effectiveness of the procedures and then produce cutting-edge results. So with that, that concludes my talk today. And so I really thank you for your time and attention. And I know we're short on time, so I am happy to make myself available to talk to you as long as you want for questions, and let y'all go. So thank you again for your time.
Are we all live? All right, cool. All right, everyone, welcome back to the final session of FlowTex 2025. It's been an exciting couple days. We've had a lot of great talks, and it's going to be summarized with 2 more great talks. So I'm really excited. First, we're going to have my staff scientist, Dr. Paul Porter, come in and give a talk of overnight staining and summarize what's in a little bit of the paper, as well as contribute a little bit of our own data, and then we'll move on to our final speaker of the day. There is a slight change in speakers. Dr. DePavia could not make it due to flu, and she did not want to get any of us sick here, and we appreciate that. And so her graduate student, Katie, will give the entirety of the talk, which will probably be better anyways. All right, so just so you know, if any of you, for those who don't know Paul, he attained his PhD in pharmacology and toxicology at the University of Louisville. And then he came to Baylor College of Medicine, ironically, just before I did, in 2006 for his postdoc in immunology. And since then, he was in Dr. Corey's lab, doing a lot of fun things, including teaching mice how to smoke. You should ask him about it. And vape now, I believe, as well, or he's working on it. And then as well, then he came to work for me, where he's been my staff scientist, and he's been the manager of my high parameter resource lab for the last 7 years. And so without further ado, Paul.
Hi, everybody. I was asked to review an article which I found extremely practical, and which is the kind of articles I really love to read myself. That article is doing more with less, improving high parameter flow cytometry through overnight staining. And I want to talk about the observations before we even get into this paper. And so the observations here, they saw an overall increase of positivity in their positive signals. And they saw this for not just surface antibodies, but they saw a vast improvement for intracellular or internalized targets using different methodologies. And one of the ones I also thought was most important was actually the improvement with the low affinity antibodies, such as like CD25. Okay, now that's wonderful to get a better signal, but they achieved that through a reduced usage of the actual antibodies. And so whenever you reduce antibody usage, you will get lots of beneficial things, such as you get a lot less antibody nonspecific activity. You see less polymer dye reactions between themselves. And we also see less of a problem with tandem dyes reacting with macrophage. And I'm going to be showing all of this data. And that's going to, of course, if you lower that background, you get improved resolution at the same time. So, and another great benefit is that you're going to see a decrease in the cost that does for you to do your panels. And so I'm actually giving a talk today that I found out today when I was researching the authors of this particular paper, that one of the authors, Oliver Burton, had already done this exact talk and put it on YouTube. So if you guys want to go to there, you'll see the actual person who provided the first part of the data we have today doing it verbatim. So I'm just gonna essentially be regurgitating a little bit of his work for you guys, but I think actually this is probably one of the most important papers I've read in the last 3 or 4 years because it's so practical and it affects everybody who does flow cytometry. And so in their first thing, they were wanting to see whether or not they could get better resolution of different populations, and a good antibody that they chose or marker detect here was CXCR5, and it was on PE-E-Fluor610. And they actually stained some viable mouse splenocytes. Now, when you go through the paper, it's actually hard to figure out what you're looking at when you're reading it. So I try to make it a little bit more simple in these slides by telling what they did. So when they started off, they were using a 1:50 dilution. Okay, and you can actually see, does this show? Yeah. You can see that you can actually see separations at 15 minutes of exposures at a 1:50 dilution of this particular antibody, but it gets better as you go up to like 60 minutes. However, when they went to like 16 hours, 20 hours, 24 hours, we had already hit a maximum for this particular antibody. And that's great because this dilution was actually set up to do a quick study. So they were actually aiming at 30 minutes. So it works great. However, they found out if they used the 1:500 dilution, they were actually able to see separation at 60 minutes, yes, with their surface antibody. However, whenever they went to these overnights with 16, 20, or 24 hours, you could clearly see that they got a lot more of actual antibody signal versus the background. So you see up here at the 1:50, the background stays exceedingly high all the way through all of these connections. However, when you use 1/10 of it, that background drops dramatically, and you have a much more clear separation of these things by using an overnight staining with less antibody. So, whenever, as a flow cytometrist, we of course had to do some stain index. And so I just wanted to show you the graph from there where they did the stain index for the 1:50 for the overnight overnight on the right, and then the short term on the left. As you can see, the 1:50 peaks out about 60 minutes like we just saw a few seconds ago. However, it doesn't actually improve much as you go further and further in time. However, whenever you're using the 1:500, you see better separation for a stained index. Admittedly, it's not a huge increase from around 12 to 15, but still, it is a good increase. And this is actually due to that reduced background as far as I'm concerned. Okay. They also wanted to do the same thing, but instead of just like a standard surface marker, they wanted to use something that was a known low affinity antibody. And so for this particular study, they used human blood cells again. They did the RBC lysis, and then they took the viable cells and they stained with CD4, CD3, CD25, CD127. And of course, they did the 30 minutes, and then they did 16 hours. And shown, of course, here is only the CD3, CD4 positive cells, because that's usually what your Tregs are that you're interested in. And when they did the 30-minute incubations, they did not see very many of your resolvable Tregs in their populace. However, when they switched this over to doing the overnight staining, without changing this to be more titrated for overnight, they clearly saw a vast increase in the ability to see their Tregs. So I personally thought they did an excellent job of this. And once again, down here, you can see their stain index for these particular populations. And of course, they had much better results with using 60-hour or overnight incubation. So in addition to just having a low affinity between the ligand and the antibody, another thing that is of concern are intracellular markers. Intracellular markers, you have to go through the cell membrane, then you got to go into the nuclear membrane in order to actually get to your point of detection if it's transcriptional transcription factor. So here we have FOXP3, which of course is that, and then we also have IL-2, which is just the intracellular ones. And so whenever they did this, they once again they got down to the T helper cells, and this time they used mice cells because they wanted to show not just human but also mouse. And what they've done is they've either stimulated with this 4-ball ester and did ionamycin with profeldin A, So unstimulated is over here, stimulated is over here. And you can clearly see that whenever they look at the stimulated version for only staining 30 minutes, you do see IL-2 production. However, it's much better if you did the overnight. And they also did the same thing, which is much more impressive, is the FOXP3, which is a notoriously hard antibody to get working, is for 30 minutes, you can see the difference. It's resolvable, but it's vastly resolvable when you start using the overnight. And once again, you'll see that this, although they have a changed thing here, it's mainly because we have a less background to begin with for the FOXP3. So, and of course, here are the stain indexes for that particular study. Once again, the overnight incubations drastically improved their ability to detect FOXP3, and it also improved IL-2. So another aspect that they covered in this paper was resolving antibodies or getting a better a signal out of internalized antibodies. So they use the classic one. Actually, they have two of them here that are internalized. The classic one being CCR7. So CCR7, when it's engaged with like it's ligand 19 and 20, no, 19 to 21, it instantly internalizes. And so for the most part, if you're looking for CCR7, you can't readily see it. And so what I've shown here is essentially it's once again the mouse splenocytes that are being stimulated, and we're comparing 30 minutes versus overnight. Now this is the surface staining. So even with the additional time with just doing a surface stain, not actually permeabilizing the cells, you can clearly see that there's an increase on for detection of the CCR7s that remain on the outside of the cells. However, whenever you take that and you then move it to an overnight and also do an intracellular staining, you can vastly see the improvement in CCR7 detection using this particular method. You're not going to see this with the 30 minutes, although they did not put that into the paper. So once again, I just make sure we show the stats. We clearly see that we can find a good titration level for our CCR7. So I don't want to spend too much more time on this. The paper's out. You can watch the video. Oh, they also did a large study on overnight staining for batch effect. So in this case, what they did was they took human blood cells and they thawed them out from the same donor, same tubes, okay? And they thawed them out 3 different times, and then they did the analysis. They did it at 30 minutes, and then they did it at an overnight. Whenever you're thinking about a 30-minute incubation, how much time it takes for you to get over to the centrifuge to get the current stain off, or whether or not someone phone called you while you were actually just about to stop your incubation, you can actually end up with variations. And you can see that there's higher variation in 3 different experiments here that were done at 30 minutes. However, whenever they did 16 hours or overnight, the kinetics already finished. They had reached their point of maximal interactions between ligand and receptor a long time ago. And so you clearly see that the batch effect virtually just disappears. I mean, it's rare you see data that lines up so perfectly. And of course, they did the comparisons of the MFIs, and there was a statistical difference between MFIs and how much they actually spread in their own right. So they actually did that for an eosinophil marker called Siglec-8, and they also did it for CD123, and they saw very similar results. So one of the things that I really loved about this article is that you could actually improve your longitudinal studies and such by actually doing these longer incubations so that there's less variation that's due to just simply how you handled it on a different day. So that kind of covers that. Next, they actually, in another study, they took human blood cells. They removed the the human blood, they removed the blood cells, and then they stained them for either 30 minutes or 16 hours. And this was a 17 fluorophore panel. Then they made a t-SNE based upon these particular markers. They selected certain ones. And then, of course, they had, on top of this is overlaid just the FlowSum cluster sets, okay? And so this is where they had taken 3 different bloods out, same blood, 3 different times, and then they did it, side by side on each of these experiments. And as you can clearly see, certain populations just utterly disappeared. And between the 3 experiments. And this led to a huge batch. This is, once again, a description of a batch variation that you can avoid by doing an overnight staining. And so if you look at the 16-hour ones, they're virtually identical looking at all 3 of them, which we kind of expected from the previous slide where we saw that they were just lined up like perfect. Okay? And of course, here are the stats that they provide in the paper. And as you, this is actually a cross entropy study. So they had a specialist come in and look at all the different flowsome groups and then analyze that and come up with like a variance version. And this is the term they have. Unfortunately, they did not describe it very well in the paper of how to actually do the math. Otherwise, I'd be happy to walk you through it. So we see a vast decrease in the chaos that comes with batch-to-batch variations. Next, they actually did another study. This one was done with mouse splenocytes, and was done at 16 hours for all of these. So this doesn't have a short time or short time versus overnight. This is just all of them are overnights. Okay, these are 23 color panel, and then they did the same 4 mice on 4 different days. So essentially, they froze them and they took out the freezes 4 different times. And then they showed all of then they looked at the individual Tisney graphs. So this is one of the mice. As you can see, they're exceedingly similar. And so what they did was they did that whole cross entropy analysis, and they found that they saw more differences between the 4 individual mice from the same mouse strain than they actually saw from the mice from day 1 to day 2 to day 3 to day 4. And so they just wanted to show in a way that the batch effect is dramatically dropped, even with respect to the actual differences between 4 different Bowd-C mice or C57s. So just they wanted to add a little bit more to the batch effect thing. So here's where I personally found this paper exceedingly useful, is cost reduction. So this entire time we've been talking about how they've been able to reduce the amount of antibodies by doing an overnight staining. And another aspect of that that's not brought out in the paper is if you're using less antibody, you don't have to switch lots nearly as frequently. So in most of the slides earlier, that we saw a 1/10 reduction of the amount of antibody that was required. And so they actually did a nice analysis, I thought, which was to take 439 different antibodies that they had titrated to be at the optimal amount for an hour-long incubation. And then they did titrations for 439 of them for doing overnight. And then they found out how much they would have in cost savings per antibody. And so the cost savings essentially went down from 23 cents per antibody down to 5 cents. And to give a little bit better feel for that, they actually did another analysis where they took 8 panels that were 23 to 50 different fluorophores. And then they found that the overnight savings was over like $13 per sample. And so when you're doing like 20 samples, this really adds up. In the YouTube thing, they had it was like 130 something pounds they found for I think it was OMIP 69 saved by just doing overnight overnight incubations and then redoing the titrations. All right, so another aspect that comes into play is here. Apparently, I got a little bit mixed up in order here. They also wanted to see what happens if you looked at low expressors. So here they've taken viable mouse splenocytes and they've labeled them for CD3 with BV570. So normally when you're doing these high parameter panels, you have less ability to pick and choose the high intensity fluorophores to go with your low expressing proteins. And so what they've done here is they've taken CD3, which is a moderate express protein, and then mixed it with a really relatively poorly bright or dim fluorophore. And then they've done it at 100, dilution of 1 to 100 and 1 to 1,000. And then, of course, they did the surface staining at 30 minutes to overnight. And so here's the 1 to 100 dilution. As you can see, at surface 30. At 1 to 100, it works. You could resolve our populations. However, if you do it overnight, you get a drastic increase in the positive population. But if you do an intracellular, you actually can increase that even more. Now, the reason that's true is because CD3 is also internalized once it's actually activated. So if you ever use PMA onomycin, you can pretty much guarantee all your CD3s are going to be inside your cell. Okay, and then they took the same stuff, and then they took the same 100 versus 1,000 dilution, and then they did it. Did it overnight, or they took the 1 to 1,000 dilution. They did 30 minutes, and of course they couldn't resolve anything because it was too little antibody for such to get the kinetics, the working well enough. Then they used it overnight, and then and which they were able to resolve it. But then of course the best one we got was with the intracellular overnight. And so it was able to pick up those CD3s that had managed to go inside the cell. And you'll notice that the thing that impresses me the most is the drastic reduction in the background. So that's really the biggest benefit is of reducing your antibodies just to get your background down. All right. This is another example of how you can start using low expressing proteins with dimmer fluorophores. And so in this case, we've stained mice cells and mouse splenocytes. And we've done that, of course, with TBET being intracellular, we did intracellular staining for either 30 minutes or overnight. And we used a 1 to 10 dilution or a 1 to 100 dilution. As you can clearly see, I'll just cut to the chase, that if you have an overnight with a greatly reduced amount of antibody used, you have a lot better resolution of your TBET in your NK cells. And okay. Another thing that you can improve is the issue that we have with a lot of polymer dyes. So as you know, all your BVs, BUVs have these polymers that love to aggregate together, and you have all this crosstalk between them if you use them in multiples. And so shown here is they've actually stained with CD3, BV570, and then they've isolated T cells for this particular study, and then they've looked at both CD8 and then Ly6G. Okay, and once again, all three of these are brilliant violet family. And so whenever you're doing this, if the manufacturers have made dye stain buffers that help with this polymerization, but if you can clearly see here that it's not really complete. So if you were to instead of, they actually use 1 to 100 for both of these particular, for these two panels. However, if you drop that, because you can now do that because you're doing overnight staining, you can reduce the amount of antibody used. And of course, if there's less of those brilliant violets around each other, they can't aggregate as well or nearly as fast because you've lowered molarity and thus the kinetics slow. And so now your brilliant stain buffer is vastly more capable of doing its job, and you'll have much more clean data. So another thing that I really liked about this, because I know that whenever I'm running at least 30 fluorophores, I'm using a lot of BVs or BUVs. All right, so they clearly showed that was very helpful. They also wanted to look at an older topic, which is not the BVs, but essentially Cy5s used to have problems with sticking to monocytes. And so what they've done here is they've taken mouse splenocytes and they isolated cells that are F480 positive. And then they looked at two different markers that are not expressed on macrophage, in this case NK1.1 and the TCR beta. And so essentially these should be completely and utterly barren of signal. However, whenever we look at them, we can clearly see we do have signal. However, if you notice, the gray ones are where they're not doing the overnights. They're using the high amounts of antibody for a short period of time. We see that they're more shifted to the right, indicating that there's more problems with background signal, because these, like I said, these do not express these cells because these are macrophage. And so whenever you reduce the amount of antibody in there, of course, once again, you're reducing the background. And so that the problem that you see with the Cy5s disappears whenever you're using this particular method. Once again, it gets a lot less complicated when you go to unmix your data later on, because you have a much more clean background to start with. And I don't know you, but I really don't like unmixing errors. All right. In the paper, they also show that cell treatments can actually alter titers as well as flow cytometrists. We all know that, but I think they did a pretty good job of showing that. So once again, we have viable mouse splenocytes. this top row essentially are the unfixed cells, while the bottom ones are the fixed cells. Okay. And so, as you can see, using 1 to 200, we see, sorry, 1 second. I'm sorry, the bottom one is overnight stained and also FOXP3 and permed. I'm sorry, this one's was a little bit more complicated. So the top row is unfixed and it's only a 30-minute stain. And of course, we see that the 200, 1 to 200 dilution of the CD3 antibody gave a decent resolution, but the 1 to 2000 does not really have enough signal to separate the two groups very well. However, if you do this similar thing down below, where they actually just did 1 to 200, and then they went to, instead of just 1 to 2000, they went to 1 to 10,000, you see a vast improvement of the signal with this one. So even though these are very similar stains, you see a much better response whenever you look transcriptionally inside of the cells. Now, this is only 2 of the different types of cell treatments that they used. I don't want to go into it any more than this. So what they did was they actually selected, what the heck? Somehow my slides are really out of order. Okay, well, we'll come back to that. So I also want to talk about a titration example for that they did for this cell treatment. So here, essentially, they're just going through a straight titration. Okay, in this case, we're looking at they're using the treatment of intracellular staining, which is the FOXP3 transcription factor kit, and they've stained all of these overnight, and they've actually selected the CD3 positive, CD4 positive cells for gating, so you can see it, and they've also done 1.1 staining, but it's actually supposed to indicate FOXP3 because it's actually expressed on an iris. So, both proteins are made at the same time, since FOXP3 is kind of a nightmare to get to show up sometimes. And they wanted a much clearer signal for their study. And so we have the obvious thing where 1 to 200, we actually see extremely high positivity. But as we keep going down and use less and less antibody, what happens is that nonspecific staining comes down and further and further and further. Now, when you're doing this with 30-minute stains, we had to use the 1 to 200 in the earlier part of the talk or early part of the paper in order to be able to see any difference. So, and as you can see here, 1 to 200 works 30 minutes surface, but whenever you're doing overnight, you can use, let me see, is that 100 times less antibody, and you get better data, and also it's a lot more cheap. All right. So comparison of the various treatments of the cells. So in this set, we have viable mouse splenocytes, and they've been overnight stained. And I'm only using one of the actual antibodies just for the description. And they actually use 6 total treatments, but shown here is only 2 of them, which essentially what we have is just cells that are stained for CD11B. And we can clearly see that, hey, those that had the FOXP3 fixed treatment worked great, not so much for the unfixed cells. So, hey, that's nice. But they also did it with using not just the eBio's FOXP3 fix and perm set. They also used the eBio's intracellular fixation kit, ICs, on this graph. They also used 0.2 formaldehyde. They used 2% formaldehyde. And then the BioLegend True Nuclear Fix stains. And then these are the results. Of course, we found out through this, once again, they're showing the stain indices as a one response thing. And they actually found that, it's 0.2% of formaldehyde, and the FOXP3 fixation kit actually worked best among groups, as that's actually these 2 up here. And so this article gets actually kind of complicated, and I feel sorry for them for having to do 6 different treatment groups. Then on top of that, doing how many titrations earlier, and then doing at different times for like 30 minutes versus overnight. The amount of work that went into this article is actually very impressive. And I've actually cut one where they started looking at the effect of if you change up the materials the liquid you stain in, instead of like you just doing one staining buffer, they did a whole bunch of them. So like I said, the amount of work. Oh, and by the way, that was just CD11B. This is actually 6 additional ones they have in the paper. I don't really want to go into it, but as you can see, each one of these different treatments has a different optimal titration value that they listed for this. So for every one that you're seeing up here, they probably did 6 or 8 different titrations or experiments. It's amazing. So once again, I want to go over the benefits of the overnight incubation methodology. Once again, the surface, intracellular and internalized targets, you can see them a lot better. The low affinity antibodies, the ones that take forever to reach their maximum. Much better because you gave them enough time to get the job done. We have a drastic reduction in antibodies. Most of these were like 1 to 10-fold decrease in antibody. You see a drastic reduction in the background through there being a lot less antibody nonspecific binding because you've used less antibody. You have less problems with your polymer dyes, and you have less problems with tandem dyes sticking to your macrophages. And of course, that all leads to better resolution and less antibody lot variations as well, because I personally find that to be very important because I'm running an antibody bank, and whenever we run out of an antibody, we have to make another lot of antibody for somebody, and there can be small changes there, so you don't have to retitrate. If that tube goes 10 times further along and getting me through a large experiment, I'm going to be super happy. And of course, there's cost savings that come in there. Now, one of the things that wasn't covered in the paper, but the reason that I fell in love with this one was because it improves our workflow. It's not just for me, it's also for my clients. So currently, if I'm doing a mouse immune phenotyping for a client, they have, the previous day, I have to do like 2 hours of work because the next day, for me to get like a spleen or a lung or a tumor, I have to prepare the suspension, then I have to do the staining, then I have to do the data collection all in one day. And that's at least an 8.5-hour day, usually around 10. And so needless to say, I'm exhausted, but it's not just bad for me because I'm getting home, I'm getting home around 7:30 at best. But my clients have to bring their actual tissues to me at 9:30 for me to get home at 7:30. That's just not nice to do to them because one of the people we worked with before we had this overnight thing, they were doing myocardial infarctions in mice. And so they had to have the surgeons in there tying these things off. So they would be here like 5 a.m. in the morning just to bring me the samples so I could actually do their analysis for them. However, now, if we do the overnight staining, we can break those days up. And so from my perspective, my first day is only 4.5 hours to 6 hours before I get to the overnight staining, and I can go home, take it easy. And I can actually do the prep work in the morning, like from 10 a.m. to 12, so that they can bring it in at around 12. And on the second day, it's a lot easier for me as a core person because I really only have to have like three hours to finish out the staining, just wash it out, and then do my data collection. And so it also makes it much easier for us because that means I can probably get that work done in the morning whenever our instruments aren't very busy. Because usually when people have to come in, they don't want to come in before 8 and do their work on a flow cytometer because they had to do 3 hours of work before that time. And so, but the benefit to the clients here is now they can bring their stuff to me at 12 p.m. and I can get home at a decent time and see my kids. So, like I said, I wish they didn't bring this particular aspect of the overnight staining out. But if you're in a core facility or if you're just actually any researcher, this is a godsend whenever it comes to your actual life. All right. I've never been one to just to read a paper. So last week I decided to try the overnight staining with mass cytometry. And so I had some the human direct immune profiling assay kit from Standard BioTools just sitting in my fridge. I was like, you know what? I'll try using that. So what I did was I took some human PBMCs we had thawed them up. I did 3 million per sample. And then I did the iridium and I did the live dead indicator, which is cisplatin 198, at the normal amounts. So I could actually see those signals. But all the rest of them, not necessarily. I would then take those cells that are thawed. They're now stained at this point, for live dead. We do FC block, of course, because I'm not a savage. And then what I did was I took the DIPA, or Deep Immune Profiling Assay kit, and essentially I would do as indicated, which would be a 1-to-1 dilution. And then I did 1 to 5, 1 to 10, 1 to 25, and then 1 to 50. And then I did it at 30 minutes. And I also did it at overnight, which actually ended up being around 20 hours. Okay. The pre-data processing I did for that was essentially we had normalization beads in mass cytometry. I removed those from the data, and then I got us down to viable singlets. And then we did the analysis. Now, there are 30 markers in here, but I'm going to try to speed through this. Okay. So shown here essentially is, since we showed CD3 so many times earlier, is the CD3. And on the left of this panel, we did is the 30-minute exposure. And this is the 1-to-1 ratio. So what the manufacturer suggested. Okay. And then on the right is the overnight exposure. And as you can clearly see, the positivity did increase with going out overnight. However, unfortunately, so did the background. And that background actually did not really get back to normal, seemingly, until at the highest dose. So that became, and that was not just once that happened, it actually happened quite a bit. And so here are all of the other ones. And as you can clearly see, oh, this one improved with overnight, that one improved with overnight, that one improved with signal, but once again, backgrounds got higher. So there's a fundamental difference between fluorophores and then mass cytometry. And it's essentially, it's how the metals are attached. There's this huge polymer that we embed the metals into to conjugate to the actual antibodies. And I have a feeling that large polymer is just randomly sticking to stuff. And it actually seems to be less of a problem at 30 minutes than it is overnight. And I have a feeling it's because it has low kinetics. So it's taking a long time for it to reach its maximum. So this is actually kind of similar to the idea of like how the tandems actually got stuck to macrophage. And I'm curious whether or not, or how the different BV or polymers actually in the BV series are aggregating. I'm curious if we could convince Standard BioTools to actually come up with something that allows, that less and less of that excessive binding. So even if we're like just blank polymer, I have no idea. But unfortunately, it doesn't look like that we're going to be able to do overnight staining for our mass cytometry stuff that I do. And so once again, we have a whole lot of these that we did. And we saw consistent things across all of them. These slides are going to be available if you actually want to delve into this. There are a couple of them that got worse. Let me see if I can find one. Not popping out. Oh yeah, here's one. This one actually was higher for signal using just the standard 30 minutes as opposed to the overnight. So it's not like there's a set rule that overnight always improves your data. So that's one of the things I worried about whenever I was reading the papers, that they kind of cherry picked certain and only showed certain markers, that entire thing. The only thing that was more broad was whenever they're using those large t-SNEs. And you don't have enough granular data there to actually see those different, if which ones were better, which ones are worse. But overall, it really didn't get much worse, not appreciably. But once again, we're looking at just random plots here. So of course, I wanted to look at something else, which is essentially a stain index. But unfortunately, I really couldn't use these index. So I'm sure we covered stain index on day 1, I believe. Okay, but the only thing I want to show here is essentially they use 2 times the standard deviation at the bottom. Okay, now unfortunately, we really can't use that in mass cytometry. If you look at this data, you'll notice that we can never really go past 0. That causes a huge problem with doing standard deviation because you don't have a standard curve. Okay, so I actually had to come up with something because I wanted to be able to analyze this data for you before I got here, and so you wouldn't have to try to look at all of these different plots individually. And so what I did was I decided that instead of using the true standard deviation at the bottom, instead I'm going to use the 95th percentile of the negative population. So in the standard stain index, essentially you're wanting to look at 2 standard deviations, which essentially would get you to 95% of the total population in your thing. So instead of using that, we're going to use what's called almost like the robust standard deviation, which is a certain percentile. I switched it to that. But I also ran into a small problem that sometimes this data is so far down here, the 95th percentile is actually close to 1. I mean, close to 0. And what happens whenever you take anything and you divide it by 0, you get infinity. And so anytime that we got to those lower numbers, it threw all the sensible data out the window. And so I decided that I would set a minimum of 5% in order to make these things. So whenever I was going through that data earlier, essentially, if I could separate it into a positive and negative, I did a stain index. If I couldn't, you really can't do a stain index on it. You know, shoulder populations, you can't do stain index on. It just doesn't work well. So instead, so of the 30 markers that are in this deep immune profiling assay, 22 of them were able to be studied. Here's an example of CD19, just showing you the actual curves and then the results that we got for the modified mass stain index. And clearly, for this particular marker, we saw a vast improvement by using overnight staining. And I kind of selected it because of that. It was one of a couple that were vastly improved by using overnight staining. And the reason is because you can see it clearly plateaus as a maximum here. However, at 30 minutes, you clearly didn't have kinetics to reach that maximum, which is here. Anyway, so from now on, you were actually only going to be looking at stain indices. And so here's the first 8 sets. And the black bars represent the 30-minute stains. Green bars represent overnights. Okay. And for many of them, I think Standard BioTools did an excellent job of doing their titrations, because you'll see that what their manufacturers suggested is actually usually the best. And so, but there are occasions like CD16, CD19, of course, CD20, we actually saw better improvements with the overnight stains. And once again, just more of those particular markers. We saw these things. I don't want to go into this directly. I will go over the graph though. So of the 22 that we were able to do stain index on, 14 of them were actually better using the 30 minutes. And only 8 of them were actually improved by using overnight staining. So my results are completely and utterly different for mass versus what it was for fluorescence. And then once again, it's because of that high background that we saw pretty much at any time we did an overnight stain. Okay, the highest median value for the positive group was actually higher in 10 of our overnights and 12 of them that were in the 30-minute stains. So once again, like I said, they did a great job at Standard BioTools getting this particular things titrated. And now, did we actually see any improvement if we used a lower dilution in the any of these markers? So essentially, remember how I said before, for the fluorescence, we were able to see they could use 10-fold or 1,000-fold decreased amount of antibody and see better results. Well, I only saw better results by using the 1/5, 10th, 25th, or 50th dilutions in 3 of them, actually, for the 30-minute exposure, which is what the manufacturer suggested. So they could have titrated 3 of them better according to what my data was. But still, that's fantastic. And there was not much difference between those 3. But for the overnight, we saw 6 of them that were vastly improved, or you could use less antibody and get better data. So here's actually the populations where we saw no clear positive, negative, so we couldn't do the actual stain index. Of course, CD45 in a blood sample, then it was this PBMC, so there was no negative, so I couldn't do anything there. CD38 was just too much of a smear here. I couldn't separate groups. And these are not the very interesting ones. They were pretty boring. We did actually find that the overnight did vastly improve the TCR gamma delta detection with the overnight, while the 30 minutes completely failed. So this is actually one of the few markers that I would definitely say was vastly improved with this overnight treatment using the DIPA kit. And we also saw an increase in CD123. Okay. And so for overnight staining in mass cytometry, the overall positivity increase was only in 16 of the 32 markers. We saw that reduced antibody usage was only really useful in 7 of the 30 markers. The background overall increases dramatically with overnight staining. Like I said, I think it's actually that polymer that the metals are embedded in. that actually causes the problem. I might do some study on that if I get money. Whenever I looked at the stain index, we only saw that 8 of the 22 markers had improved with the overnight staining versus just the standard 30-minute things. So the conclusion, of course, is though overnight staining improves the signal, the impact is minimized because the background increases so much. And that kind of concludes what I had to say today.
In sake of time, we'll take questions at the end of both talks so that so we can make sure to get everyone home on time. All right. Now for our final speaker, we have Katie Sholand. Thank you.
How long has he known me?
Hey. All right. Katie is a 5th-year PhD candidate at Rice University in Dr. Cintia de Paiva's lab. Her thesis involves the investigation of pathogenic CD4 T cells in the role of autoimmune-related dry eye disease. Her interests include autoimmunity, Sjogren's disease, age-related dry eye, and identifying therapeutics to combat autoimmune inflammation. Previously, she was an undergraduate intern at Sandia National Laboratories, where she worked on several projects, including the development of assays to diagnose hoof-mouth disease in cows. She has won the Samuel Wu Award for graduate student research, the Gene Anderson Award for Best Paper, presented at the OU Vision 16th Annual Graduate Student and Postdoc Fellow Vision Workshop. Wow, that's a long one. All right. And she did. We're almost done. All right. And she graduated summa cum laude from the University of New Mexico in May of 2020 and is accredited biochemist by the American Society of Biochemistry and Molecular Biology. And most importantly, she is one of my favorite partners next to my office because the Canto she uses is right outside my door. So I get to every once in a while take a break and have a little chat. So without further ado, Katie.
Thank you, Joel. Joel is also an avid Lord of the Rings fan, so sometimes we talk about that while we're doing Flow. Good afternoon, everyone. I am your last talk, so hang in there. So thank you very much for hanging in to the very end. Thank you very much to the organizers. In case you can't tell, I am not Cintia de Paiva, well-respected and well-known professor in the Department of Ophthalmology. I am her graduate student, Katie Sholand. She has entrusted me with this talk as, unfortunately, she has been bedridden for a week with the flu. So everyone, please take care out there. Something is really going around. So just a quick disclaimer, we stole many cute cartoons from cellcartoons.net for this presentation. We love using them in lab, and we are happy to use them in this presentation. We are an ocular surface laboratory, so we are very interested with the front of your eye. And one of the most important tissues in the front of your eye is the cornea. What's interesting about the cornea is it's the only transparent tissue in your body, and it's actually the most densely innervated. And those reasons are intertwined. It's because the cornea is the only tissue which needs light to be able to pass through it and to focus it onto the retina, which is how you have vision. So the cornea needs to stay clear in order for you to see. And in order to protect the eyes, we have very intense neurons in the eye in order to protect it from any sort of harmful stimulants. And your eye has to stay wet in order for you to have clear vision because you need a smooth ocular surface for light to clearly enter the eye and hit the retina. And so what keeps it smooth and wet is the tear film. And the tear film is made up of 3 components. The first is the lipid layer. If I do this, can we see? Yes. The first is the lipid layer, and this is produced by meibomian glands that line your eyelids. This prevents the tear film from evaporating off the eye, and so it allows us to have lubricated eyes. The second and biggest portion is the aqueous layer. This is produced by the tear-secreting lacrimal gland. It sits just below your eyebrows. And third, there is the mucin layer, and this is produced by epithelial cells, goblet cells in the conjunctiva. And so when you have all 3 of these working in harmony, you have a beautiful tear film, you have beautiful, clear vision. But I'm not here to talk about perfect health. I'm here to talk about disease. And so when any of those components are disrupted, you end up with a disease called dry eye disease. So dry eye is an umbrella term referring to any interruption of the tear film from any of those three components that I talked about. It is a multifactorial autoimmune disease, so it is a chronic inflammatory disease. And some of the symptoms that we see with dry eye include increased sensitivity to glare, blurry vision because your tear film is no longer stable, sensitivity to light. There can be a foreign body sensation. So if you've ever had like that feeling of an eyelash on your eye and it's bothering you, dry eye patients have that feeling constantly. They can also have a gritty sensation and unfortunately quite severe pain. With a diagnosis technique, we're able to determine that the pain is equivalent to that of angina, which is chest pain. So this can really affect the quality of life of patients. Dry eye disease patients actually have increased rates of insomnia, anxiety, and depression because their quality of life and their daily life are so impacted by this disease. So there are several risk factors with dry eye. Some of these we cannot help, such as old age or female sex. Other ones we could help, but we like using them because they're convenient, such as contact lens use and using computer screens. So everyone in here, please take a couple strong blinks. Thank you. Your eyes will thank you for it in the long run. So we have previously characterized the eye in several ways. One is the human conjunctiva. Again, that's where the goblet cells are that produce the mucin layer. And we found that there are lots of immune cells in the conjunctiva, including CD3 T cells and antigen-presenting cells. So I know people think, oh, well, it's an immune-privileged site. There's not a lot of immune cells on the eye, but actually there are, and they're there to help protect and keep your vision clear. We have done a lot of research investigating the lacrimal gland, which is the gland that produces the aqueous portion of the tear film. So I'm going to show you some of our work in the murine lacrimal gland. It's this big, juicy-looking gland here right below the ear of the mouse. So there, as I said, age is a risk factor for dry eye, and we have reported age-related changes in lacrimal glands. On the left here, you see an 8-week lacrimal gland, and on the right, a 24-month lacrimal gland. And as you can hopefully see here, there are these accumulation of lipid drops, as we call them. There's some sort of accumulation of fat in the lacrimal gland. And when you look at this histologically, what we see are dilated ducts, as you can see in this image here where the asterisks are, and infiltrating immune cells to the gland. And so we have characterized this. Again, on the left is the 8-week lacrimal gland, and on the right is the 24-month one. And all those dark blue areas are infiltrating immune cells. And so likely what's happening is the immune cells are infiltrating the lacrimal gland and disrupting the production of the aqueous tear film. We have characterized these infiltrates quite extensively. We have seen that these infiltrates contain both CD4 positive T cells and CD8 positive T cells, as well as B cells and T follicular cells. We've also done, of course, this is a flow talk. I'm going to talk about flow, I promise. We have done intracellular staining in the lacrimal gland, and what we have shown is that there is an increase in Th1 cells in the lacrimal gland. So these top two graphs here are in the lacrimal gland, and as a control, I have cervical draining lymph nodes on the bottom. And this is the 8-week mark, and this is the 24-month. And so hopefully you can see here that there is an increase in interferon gamma producing CD4 T cells. We've also looked at IL-17 producing CD4 cells, and we don't see that similar increase. So we see an increase in Th1 in aging in the lacrimal gland. So if you're interested in studying dry eye, we're interested in these units: the lacrimal gland, the meibomian gland, the cornea, and the conjunctiva. And we can do flow in all of these tissues. Unfortunately, some of them are challenging for flow cytometry, and they include the lacrimal gland, conjunctiva, meibomian glands, and cornea, which if you just paid attention, that's all of them. Also, if you're interested in doing flow in human patients, other challenging tissues include tear washings from humans and impression cytology. So this is how I felt when I was told this. So some of the reasons for this challenge is that some of these tissues can be dense. That means that they have a lot of connective tissue, which requires digestion. There are epithelial cells mixed in with stroma cells and the immune cells. So these are not clean populations like a lymph node or a spleen. We have to differentiate between them. And so depending on your digestion process, sometimes the cells can be sticky is the easiest way to describe it. It'll get like stuck in the pipette and you lose your cells. But this is a motivational talk, and I'm here to give you some tips on how you can actually process those. So just some general tips that you all know, but I'm going to reiterate. First, we always recommend doing pilot studies. Don't waste your precious samples if you haven't nailed down the protocol. The second, and this has been repeated ad nauseum, I know, at this FlowTex talk, but titrate your antibodies. We also recommend when you're working with tissues like this, which can be more sensitive, that you work fast and on ice. We recommend not spreading out your experiments. There can be batch-to-batch variability, so we recommend doing all your experimental samples on the same day. And I think probably the most specific advice from our lab is always use tissue-specific FMOs. Gating from a CLN FMO is very different compared to gating from a lacrimal gland FMO. They just behave differently because of the mixture of cells involved. So for the rest of this talk, I'm going to talk to you a little bit about how we prepare these difficult tissues for staining. I'm going to talk a little bit about overnight staining, but I feel like Paul really knocked it out of the park, so we're not going to talk too much about that. And finally, I'm going to talk about how we are able to actually freeze these samples despite them being kind of tricky, and we can actually thaw them up to 2 years later for use. So when we're processing lacrimal glands, the key thing here is that we use something called collagenase 4 in a very small solution. We excise them from the mouse and incubate them with chop after they've been finely chopped for 60 minutes at shaking with 37C. In the last 15 minutes, we add DNase, and all these steps combined ensure that those glands are no longer sticky. The samples are then neutralized, filtered, centrifuged, and resuspended for further processing. Something that I want to note here is that the filter size for these tissues can actually affect certain populations. So for instance, if you're interested in macrophages, I would recommend using a larger filter size, like 100 microns, compared to 50 microns. So our lab typically varies between the 50 micron and the 100 micron, depending on the panel and the cell populations of interest. The conjunctiva is another one of those tricky tissues. We are able to excise 2 of them per eye, which means per mouse we get 4 pieces, and we again immerse it in that collagenase 4 solution. We incubate it the same as with the lacrimal gland with shaking at 37C for 60 minutes. We then add DNase in the last 15 minutes, and then it's the same as before. We filter, neutralize, centrifuge, and resuspend. Recently, we've just started doing meibomian glands, and these have proved particularly tricky because they're so sticky. So as I told you before, the meibomian glands line your eyelids. So the best way to actually dissect them is to, I'm sorry if this is gross, you cut out the eyelids, and then under a microscope, you dissect out the tarsal plates. So here is an image of what the meibomian glands look like. Those fluffy white areas are the actual meibomian glands. And so we are able to dissect them out under a microscope. And then we put them in a solution, this time of a different collagenase called collagenase D. We found that this one is better at digesting the meibomian gland. We then, as usual, incubate at 37C for 60 minutes. But in the last 15 minutes, we also add trypsin EGTA along with our DNase. And we found that that's really improved cell volumes when we do this experiment. And then, as always, samples are neutralized, filtered, centrifuged, and resuspended. The last tissue I'm going to show you is the cornea. This one is particularly tricky because there are so few cells in the cornea. So something to keep in mind is to think about when you're going to actually chop up your cell for digestion. So first what we do is we dissect out the cornea in as intact one piece as possible. So we try not to damage it in any way. And we let that incubate in 20 millimolar EDTA for 20 minutes. This helps with the tight junction proteins on the cornea and helps for further steps, but you don't want to chop it up at this point because if you do, it's still very sticky and you will lose a lot of cells. So after this, we then take the whole cornea and place it into a solution of 0.1% collagenase 4, at which point you can then finally chop it and incubate it at 37C. But this time, because there are fewer cells, we try to reduce the incubation time, so we only do 30 minutes. And then, of course, in the last 15 minutes, we add DNase. And then just as before, neutralize, filter, centrifuge, and resuspend. When you're looking with the human samples, There's actually, thankfully, not a lot of processing to do, but you have to keep some things in mind. For example, for human tear collection, there are very few cells and the volume is very small, so you cannot freeze these and work with them later. You have to work with fresh samples, which leads into the second problem, which is there is batch-to-batch variability. You know, humidity is different on days, allergies might be acting up, and so humans have very complex immune systems, and you're trying to account for as much of that as possible, hopefully by coordinating collection on the same day. But this means it's very difficult for the doctor because you have to synchronize when patients come in and when the doctor can collect the samples. But please try as hard as you can to do these things because it really does help minimize your batch-to-batch variation. And as I said, it does not require processing, so you can just proceed with staining immediately. Finally, for human impression cytology, this uses this little device here called an iPrim. I know sometimes people get a little squicked out. They're like, oh, I don't want that near my eye, but this is very safe and very harmless. It applies a small filter paper to the conjunctiva of your eye and removes a uniform layer of cells, which you can then process. But again, this is a few cells, so you have to process them fresh, but it does work. So again, like with the human tear samples, there is batch-to-batch variability, so we recommend synchronizing collections. And particularly with this human impression cytology, we find that since the cell number is so low, it's better to use a panel that investigates abundant cell populations. Here, for example, we're looking at an antigen cell-presenting marker, which is abundant in these samples. So next, I'm going to talk a little bit about overnight staining of samples. You've already heard some of this, but that's okay. So at this point, your samples have been processed appropriately. And at this point, you can do lymphocytic stimulation, such as with PMA and iomycin, if you so were inclined. If you're not, otherwise, you would head directly into FC block for 5 to 10 minutes, and then you're live dead staining. At which point, we fix immediately. Sometimes we also permeabilize if we're interested in intracellular markers. And then we prepare one cocktail with all of our antibodies combined. We have extracellular, we have intracellular, they're all in one combination. And as Paul really pointed out in his talk, you can use a much lower concentration because you're staining overnight. We use a half to a quarter of the antibodies. We have not gone down as far as 1 to 10,000, but you probably could, which is that this has worked for us best. And then we just apply the stain overnight in the dark at 4C, and the next day you can come in and analyze it. And what I have found works as well is that this can also stain over the weekend. So I can apply the stain on Friday and I can come in on Monday and I don't have to ruin my Saturday by being at the flow core and cursing my PI. So I want to show you some pictures of this. This is just to reiterate some of the data that you already saw in that paper. Here is 2 lacrimal gland samples. They have both been stimulated with PMA and iomycin, which is why it's kind of hard to identify the CD3 positive versus the CD3 negative here. So we're staining for both CD3 and CD4. In the short stain here, you can obviously see the population, but it's kind of not as well spread out as we like. Compare that to this lacrimal gland that has had the overnight staining. The populations are much easier to identify. We also have success with this with intracellular cytokines. So here I'm just showing you our favorite cytokine in our lab, interferon gamma. So these are Th1 cells, so they're CD4 positive, and we're looking at the ones that produce interferon gamma. Again, with the overnight staining, what we love is that there's just a very distinct population. It's easy to see. It's easy for me to gate as a novice learning flow cytometry. It's just a lot easier. And we also have, as Paul was mentioning, a very tricky transcriptional factor, FOXP3. We're very interested in Treg cells in our lab. So I'm showing you here cells that are positive for CD4, and then we're looking at the transcription factor FOXP3. And again, I think this one is the most striking. It's very easy to see which ones are Treg positive, or Tregs, which ones are FOXP3 positive, as opposed to the negative in comparison to a short stain instead. So some of the benefits from overnight staining, I know I'm just repeating facts here, but they're worth repeating. One is that in our protocol specifically, because we're able to fix the cells almost immediately, there's a much less time that those cells need to be hanging around. So especially with delicate cells, this is nice. You live dead stain them, you fix them, and then you don't have to worry anymore. And so what we found is that this results in greater numbers of viable cells when we're looking downstream at our analysis. Paul showed you that great graph, so I don't have to, but yes, reduced antibodies means reduced costs. So this is great for grants because we all know that we're in a bit of bind right now. And I'm a graduate student. My life matters to me, so I appreciate time when I'm not in the laboratory. So, you know, these sort of protocols, which give more time for us to focus on other things, is very much appreciated. So I don't have to mention this paper, but I'm going to. If you would like to further read, I do really recommend this paper. It is the reason that we changed our protocols, and I would just like to thank White, Toombs, Liston, and Burton for their work. It is truly helped my work a lot, and I think improved our results quite a bit. So finally, I want to talk about how we freeze cells in the lacrimal gland and the meibomian gland, which are, again, those sort of sensitive cells, and how we can then later thaw them in a much longer timeline. So what we do for freezing is we process lacrimal gland and meibomian gland cells as we did before, so the process does not change. But after you pellet your cells, we would then resuspend in 1 ml of our freezing media. I'm not sure how different our media, I can't think right now, but it's probably a slightly different formula compared to the ones you might use. It's a lot of FBS. I feel like we use a lot of liquid gold in our lab. So ours is 90% FBS and 10% DMSO. And I have to stress here that our DMSO is cell culture grade. Whenever someone tells me that their cells were no longer viable after thawing, I tell them your DMSO went bad, and it's almost true 90% of the time. We then place our samples in a cell freezing container and place them in -80°C for 24 hours. We really like these foam ones. We call them Mr. Freezies. They're really great, but in a pinch, and if you don't have the money for that, you can just use 2 styrofoam containers on top of each other, and they work approximately as well. So then afterwards, we move them to liquid nitrogen for long-term storage, and I do mean long-term. We can use the samples up to 2 years later, and I'm going to show you some of that. But as I want to note, you can actually do PMA and iomycin staining from our thawed cells, but we recommend that after about a year, you no longer do that because at that point, they're much more fragile. So when you thaw cells, you need to pre-warm your RPMI media to 37 degrees, and you have to use 10 ml per sample. I know I'm saying a lot of expensive reagents right now, but it really does help with your viability down the line. And so our recommendation is to thaw your samples as quickly as possible in a water bath, and then place them in that large amount of RPMI, and then move them to ice while you're preparing all your other samples, and then immediately centrifuge them and resuspend them in complete RPMI, and then proceed with your staining as normal. So this is a sample that I froze in February of 2023, and that I then thawed and did overnight staining earlier this month, February 2025. This is a lacrimal gland, so this is not just a spleen or anything. That's why there's so few lymphocytic cells in such a massive population. And I just trying to emphasize here how, because we were able to just thaw them and then immediately fix them, we were able to keep them alive long enough to do really complex staining. This panel in particular was something like, I think, 25 colors. And so we got a lot of valuable data out of it, despite the fact that the samples had actually been in the liquid nitrogen for 2 years. And I just want to shout out here Amy and Claude. They were the ones who helped run this panel. I gave them 2 plates. It was something like 100 samples, and they didn't kill me. So thank you very much to the Baylor Flow Corps. So if you're curious for further reading on doing flow cytometry in the eye, we recommend these 2 protocols. They've also been helpful in modifying our own. I would also just like to thank the Baylor Cytometry Corps for all their help over the years. You guys are cool, you guys are competent, you guys are stylish, and you're never mad at me, even when I accidentally break all your machines, for which I am very appreciative. The ARIA 2 hates me. I mean, we know this. I'd also like to thank the Paiva laboratory. Unfortunately, my PI couldn't be here today, but I know she's watching me on the Zoom. So hi, Cintia. You know, we never know where the science is going to take us, but we really wish it took us here. I'd like to acknowledge our funding sources. And with that, I'd like to take any questions. Thank you. or we can all just go. You know, I know you're tired.
I'm really sorry. I have a question.
That's fine.
I actually have like a scientific question and then a like protocol related question.
I'm here for all of those questions.
Okay, so my first question is about the lacrimal gland.
Lacrimal gland, yes.
Yeah, that thing. You said that there's infiltration of lymphocytes over when they age.
Yes.
And you said that there's T cells, specifically Tregs and TFH cells, and then you said there's B cells. Have you characterized those B cells? Is it actually like a germinal center that's forming? Like what's happening?
This is such a great question. Excellent question. The answer is yes. Cintia recently published a paper looking into this. We have very thoroughly characterized those infiltrates. They are germinal center. There are B cell germinal center cells in the lacrimal gland. And so we call it actually a tertiary lymphoid structure because it's not in a lymph system, but it actually does have those clinical aggregates like you would see in a lymph node. So yes, they are there. And we think that's probably one of the reasons for dry eye.
Wow, very interesting. My technical question is about your FOXP3 staining, which
Yeah.
is beautiful with your overnight. I specifically just wanted to know if A, you knew which fixed perm kit you were using or if you're making your own, and then B, if you know specifically what clone of FOXP3 you're using.
Okay, I don't know the clone off the top of my head, but we're using the BD transcription factor fixed perm kit.
Okay, thank you.
Thank you.
Okay, we got some questions from the internet.
Oh,
Um,
they're paying attention. Blink.
okay, here's the first one. What is the viability percentage of cells isolated from the cornea using your protocol?
Using the protocol, it's about 50%. So it's doing pretty well, is what I would say. The camera's here, right? It's there. Okay. It's doing well.
And then the second one is kind of a little more general, but people are very interested in viability. So
Please ask me away.
yeah, so the question is, has the viability of the cells stayed the same after overnight staining?
Well, so we do the live dead staining first, and then we fix the cells. So the viability doesn't really change. You can't do live dead staining after you fix the cell because then they're dead and they would uptake the live dead dye. So I don't think I can answer that question. Yes, the answer. The same viability, exactly.
Well, no, it's much lower after but yeah, the way it's detected, it's good, I guess. Okay.
I just had one question.
All questions are welcome.
So I'm not an eyeologist.
Yeah.
I've heard that one of the unique features of like corneas and stuff is they continue growing throughout your whole life. And that's partly why your vision changes as you get old. You just get more and more cornea. Is that the case in mice? Like, do you get more cells in these 24-month-old mice than in the 8-week-old mice? Like, are they bigger?
Yes, they are actually. It's interesting. The lacrimal gland too, you get more cells in them when they're older. So it's actually easier to do flow in older mice because I just have a higher population. So yeah, they do get bigger and it makes flow a little bit easier. But the downside is aged mice are very expensive.
And is the greater numbers of cells, is it just those infiltrating lymphocytes or is it epithelial connective cells?
In cornea, it's epithelial cells. In the lacrimal gland, it's those infiltrating immune cells. So it depends on the tissue.
Thanks.
Thank you.
Great talk. One technical question and thought. So I noticed that you had to change up your protocol on your digestion to include EDTA, a trypsin EDTA mixture. And I was just wondering, since DNase actually is calcium dependent, and it does work without calcium, just very minimally. If you've tried trying to add the DNase a little earlier, so it would actually be maximally work, and then do your EDTA in that process, or if you saw any issues at all, because once you add the EDTA, obviously it chelates out all the calcium.
So I shortened it for to make it simpler to understand. But actually, what we do is we add the DNase 15 minutes before, and then 5 minutes before. So the DNase is alone for 10 minutes. Then we add the EDTA trypsin. I thought it'd be a little confusing if I did it like that, but yes, we did keep that in mind.
Perfect.
Thank you, Joel. All right, you guys hung out to the end. Thank you very much.
It's time for everybody's favorite part of FlowTex. When Joel stands up here. Well, actually, Rui's gonna be Vanna White, I think, today. If you're here and you filled out your raffle, please come on down. Should we spare the online watchers?
Probably spare the on.
Okay, well, for the
Yes.
people online, although I'm sorry if this is a problem, but for the people online, I think we're going to end the stream here. But thank you very much for joining us from all across in Houston and Texas, the US and the world. And we look forward to seeing you guys in 2026.